Okay, so I'm going to be talking about part of my PhD research at Bristol University, which is using molecular and isotopic approaches to investigate freshwater resource use by early farming populations in Central Europe. So research is increasingly showing the diversity of the Neolithic transition in Europe, but we do know that for the central region associated with the linear band ceramic LBK culture, it's very much based on domestic animals, generally with low exploitation of wild animal resources, but certainly some regional variation. Freshwater resources, on the other hand, so fish, but also mollusks and other shellfish and aquatic birds like ducks, generally we don't see them being recovered in huge numbers. But even at sites where the number of remains might be quite low, whether that's due to poor preservation or differences in excavation techniques, even if that's a true reflection of how important those resources were, we're still interested in kind of identifying them and understanding them to see how farmers were exploiting their local environments, and also just sort of understanding the diversity of this transition. So one of the ways that we can look at animal exploitation is using stable isotopes of human remains, but this is quite limiting for looking at low contributions of freshwater resources to the diet. So that's partly because you need quite a significant input of aquatic resources to the human diet to actually see a shift in the values, but also because we see such wide variation in freshwater values. That's partly due to species differences. So you can see for Lake Constance, we've got burbot and pike. They're completely different. But then for a different freshwater environment, you don't see the same variation. So that means that constructing a local baseline for freshwater resources is quite difficult. And if we're only including like a small number of species in our study, or potentially picking them from slightly different freshwater environments to what was being exploited in the past, then we might not be seeing a true reflection of the local baseline. So one of the other ways that we can look at animal exploitation, and what I'm going to be focusing on today, is lipid residue analysis. So I know Melanie already talked about this earlier, so very briefly, lipids get absorbed into pottery vessels during cooking and other processing methods, and then they can preserve there for archaeological timescales until we excavate the pottery, and then we can extract the lipids and analyze them, and then match the compounds that we detect back to known precursors from different animal resources. So this analysis is generally done using a combination of gas chromatography, mass spectrometry, and also GC combustion isotope ratio mass spectrometry for doing compound-specific fatty acid analysis. So this compound-specific analysis focuses on palmitic acid and stearic acid, C16 and C18, so that's just indicating the number of carbons in the molecule. And these are generally the compounds that are recovered in the highest abundance from animal fat lipids. So on the plot, you can see got the C16 on the top axis and the C18 on the side. And this is a very good proxy for separating out different types and sources of terrestrial animal fats. But for freshwater fish and migratory fish, we see a much broader range of values. And that's due to the same species and environmental differences that you see in the bulk collagen values. So essentially what this means for pottery lipids is if we've got a vessel being used for a mixture of terrestrial and also aquatic resources, potentially low levels of aquatic resources, then we might not be seeing a huge contribution to the delta 13C values coming from the freshwater, so it can be quite difficult to identify. Luckily, there are these other biomarkers that we can use for identifying aquatic resources. So fish and also other aquatic products are very high in unsaturated and polyunsaturated fatty acids. But these are quite unstable molecules. They've got a lot of double bonds. That means that they don't preserve very well because they're quite reactive. But if you heat them up, then you form AKAs, two bonds here, and then dihydroxy fatty acids are also a degradation product of those. So this is where the double bond would have originally been. And it's these really long chain C22 compounds that are most diagnostic of aquatic products. You don't really get them in terrestrial animals. And then the last group is isoprenoid fatty acids. And these are found in quite high abundance in aquatic products. And they've got a very branched structure, which makes them much more resistant to degradation. And TMTD is the most sort of diagnostic of aquatic foods. So generally, these compounds are detected in pottery from marine sites where we see quite a clear signal for aquatic resource processing. But if we're looking at those lower levels, then these compounds might be present in much lower concentrations and also potentially sort of near or at the limit of detection for the instruments that we're using. So to address this, we've been working on a more sensitive method for detecting those biomarkers. And that's using an instrument called a GC quadrupole time of flight mass spectrometer. So generally, this biomarker detection is just done using gas chromatography mass spectrometry. And we use something called selected ion monitoring mode to actually target specific masses from the compounds that we're interested in. So rather than getting a full mass spectrum, you just get the masses that you sort of told your instrument to scan for, which does work, but it could be better, basically. And depending on the compounds that you're interested in, you have to use different columns in the instrument in order to be able to target the different ones. The GC QTOF MS, on the other hand, is a much more sensitive instrument. It's more selective and higher resolution. So essentially what this means is that we can detect those aquatic biomarkers in lower concentrations than with GCMS in SIM. So it uses a non-targeted approach. So rather than just selecting those masses from the compounds that you're looking for, the instrument will scan for a whole range of masses, and then you can extract the data for the compounds that you want. So that means that you get a full mass spectrum like this, which is much better for identifying the presence of your compound than this. And one of the benefits of that as well is that we can do retrospective searching. So if we identify potentially new biomarkers, then we can go back to our data and kind of search through to see if they're there. And it also means we can do simultaneous detection of all our biomarkers within one analytical run, rather than having to do it on different instruments, which just reduces the analysis and the sample preparation time. So I've applied this method to a few sites on pottery lipids from the NeoMilk project. So I'll just go through a couple of examples to show how kind of combining the high sensitivity biomarker data with compound-specific stable isotope analysis. So the first site is Tiszazolos, an early Neolithic site in northeastern Hungary, where we see some quite early linear pottery culture material. In terms of the faunal assemblage, a relatively high proportion of domestic animals, but also quite a lot of freshwater remains. So mussel shells and also quite abundant fish remains. But the issue here was that when they were excavating the site, they didn't actually see the fish bones to be hugely important to the function of the site because it was Neolithic. So they were never actually analysed properly or sort of quantified in any way, and were just thrown out and never reported. So if we look at the pottery residue data, so it's the same plot as before, the C16 and the C18 fatty acid values, and then that's plotted against modern reference data in the ellipses. So each of the points on the plots represents an individual pottery vessel, and the orange circles are those in which no biomarkers have been detected, and then the blue stars are where we've detected just one of the biomarkers or the two most diagnostic in combination. So you can see that there's quite a cluster sort of towards that freshwater fish range, and then a few of those samples are also containing those aquatic biomarkers. So definitely there's some evidence there, I think, that the pottery vessels were being used for processing these freshwater resources. The second site is APS, a slightly later LBK site, and that's located in northeastern Hungary again. But the faunal assemblage at this site is, there's a much higher proportion of domestic animals. I think there were sort of like a handful of fish remains recovered, but I don't think the dating of these or their association with the LBK material was very certain. But if we look at the pottery residue data, you can see quite a broad range of values kind of in the terrestrial product range, but quite a few of those samples are containing those aquatic biomarkers. So I'd interpret this as potentially some low levels of freshwater processing, but clearly not enough as at the previous site to actually shift them anywhere near the freshwater fish range. And interestingly as well, there was some analysis of human remains from the site, but no freshwater contribution could be identified from that. So finally, just to compare the two sites side by side, the y-axis, the big delta plot here, that's just showing the difference between the C16 and the C18 values, just to kind of separate out the different sources of animal fats. So at Tissozolos, you can see there's that cluster in the kind of non-ruminant range up there, whereas APS, we've got much broader range of values. And those samples with aquatic biomarkers very much kind of plotting across that entire range. So obviously this is only two examples, but you can definitely see that there's a difference potentially in how the pottery was being used, and maybe with more mixing of ruminant products at the second site. So just to sum up, freshwater exploitation, especially at low intensities, can be quite challenging to identify using bulk collagen analysis of carbon and nitrogen. And those low contributions as well can be difficult to identify using fatty acid delta 13C analysis from pottery lipids. But we've been working on a more sensitive detection method using GC-QTOF-MS, and using this to investigate freshwater resource exploitation by early farming populations. And then in terms of future method development, we're hoping to establish biomarker detection limits and also hopefully discover new biomarkers that might be associated with freshwater resources. So finally, just some acknowledgements, especially to Laszlo for providing the pottery from the Hungarian sites. And yeah, that's it. Thank you very much for listening.
Hello everyone, I'm very pleased to be here today to present you my master thesis. This project is part of the INR ASAP, which is coordinated by Olga Otero. The main aim of this project was to better quantify the intensity of seasonality change in fossil assemblage. So seasonality can be described as a cyclical seasonal change, which occur in precipitation or temperature. It's interesting to study this seasonal variation in order to understand the evolutionary dynamics of species. And this variation can take different form depending on the locality, with, for example, multiple dry season or rainy season. To study this in the fossil record, you can use the community structure or the principle of actualism, but it gets you having some bias. As so, quantified by biochemistry can be therefore more interesting. As so, in this study, we use isotopic record to define the cyclical change in precipitation and temperature. And the oxygen ratio in temperate environment is an indicator of temperature, but in this subject, it's for tropical environment, so it's inversely correlated with the amount of precipitation. It's called the amount effect. We also use carbon ratio, which the record provide insight into herbivore diets due to the different split in carbon for the C3 and the C4 plants. The species used in this study was Hippopotamus amphibius, which is currently one of the one species of the two species of a very abundant family in the Neogene and Quaternary. This semi-aquatic species has a lower oxygen ratio and less variable. And they also are herbivorous with a diet majority of C4 plants, but with a proportion of C4 plants that is influenced by the season. They also have a continuous growth canine with sexual dimorphism that can provide a long-term recording of this isotopic record. So enamels was chosen because it's the response to genesis. And the long-term goal is to use the same method on the Pliopleistocene hippo. So it's more interesting. And when you take a serial sample on a canine, a problem arises, which is the averaging of the signal from the long mineralization time. This averaging will cause a dampening of the signal, but in order to overcome this problem, the model of Passy et al. Can be used. It was it used the enamel mineralization parameter to re-amplify this signal, and this model was developed on hippocanine under the assumption that the growth rate of this canine is continuous, is constant, but it never have been verified. So the main subject of my thesis was just to study the extant hippo in order to calibrate this model through isotope and dental histology. The long-term goal is to apply this parameter of mineralization on the enamel of fossil specimen to reconstruct the past seasonality. So for this project, we had 3 individuals, one male juvenile, one male adult and one female adult. And I've proceeded to do histological thin section from all these extracted teeth, and the objective was to observe the internal enamel structure at a microscopic scale. So using stitched image of thin section, I focus on the increment line. You can see here. It's the deposit of enamel, and they were counted within each 5 mm segment. It allows us to obtain a rate of enamel extension in micrometer per increments. And in ungulate, it has been related to circadian rhythm for the brachionodont and ipsodont molar teeth. And it can help to provide an estimate of the growth rate of the teeth. So this extension rate by individual showed interesting result. You can see here. On this graph, there are you can see them as box plot attributed to each individual. The x-axis is for the different individual, and the y-axis are the enamel extension rate in micrometer per increments. And we can observe higher strong inter-individual and intra-individual variation. For the juvenile, you have a higher extension rate than for the adults. Stronger extension rate for the lower than the upper canine on the juvenile, and no difference was seen between the male and the female adult for the upper canine. I've decided to present you more the juvenile of this study. On the specimen, we took the front molar, the lower and the upper canine, and we proceed to do histological comparison. Note that the front molar was unerupted at the time of the death, and it was still in the maxillary bone. As so, we proceed to do isotopic sample on the front molar and the upper canine, with the assumption that the cervical part of the upper canine is corresponding to the front molar. So this gives us this comparison for carbon and oxygen. You can see the carbon in red and in blue the oxygen, and in the light red and blue the data are for the molar, and in dark red and blue is for the canine. You can see that the isotopic variation show a pattern corresponding to seasonal cyclicity. And according to the amount effect, the high value in oxygen ratio corresponds to dry season, and the low oxygen value corresponds to rainy season. The curve showed similar shape for the canine and the molar, but the range is different due to the different degree of signal dampening. From those data, we consider that we have a record for about 1.5 years for about 60 mm for the canine and 45 mm for the molar. And when we compare this to the dental histology, we counted for the canine 320 increments, which leave us with 0.6 increment per day for the canine, but for the molar we counted 500 increments for this distance, and it leaves us with 0.9 increment per day. So you probably have a difference in the periodicity of the increments between the upper and the upper canine and the molar for this specimen. And we also have studied the adult Chad, which is a little more interesting because we know more about this individual. This death was in March 2006. Sorry. And the isotopic record can therefore be interpreted with the knowledge we have of the environment where it comes from. So the specimen lived in the Manda National Park near the Chai River. This river had its source in the Central African Republic, and this locality have a tropical seasonal regime with one rainy season per year and a strong amount effect. The isotopic data of this canine is presented in this graph. In red, you can see the carbon, in blue, the oxygen isotopic ratio. And the isotopic sample shows cycle corresponding to the different dry season for the high value and the low for the rainy season, as for the other. And these two variables are correlated and follow the same periodicity corresponding to the season. They also have a slight lag between them. And this indicates a higher consumption of sea-free plants during the dry season compared to the rainy season. And the part of sea-free plants in the data is far from negligible and could have reached more than 50% during the rainy season. It is congruent with the previous study by Souron and his colleague in 2012, and it confirmed that the common hippo is best characterized as a seasonal mixed feeder and not a grazer. When we add to this data, this isotopic data, the histology study, we observe local variation in extension rate. And as for the other canine, and we can see that they vary strongly within the life of the individual between 60 and 220. And on our chat canine, the seasonal recording represents 7 seasonal cycles, which means we have a recording for the canine of about 6.5 years. The number of increments counted on the chat canine was 622 increments. As so, the average periodicity for this canine is 0.7 increment per day, which does not correspond to the other ungulate in the literature. And it is comparable to the estimate 0.6 increment per day of the juvenile hippo. This variation in the rate of increment produced daily is quite discussed in other species as rodent or primate. So we wanted to know now if for the model of Passy and his colleague, it had an impact that we have a different mineralization parameter. So as this model is based on a histological parameter and sample geometry, we have one of our collaborators, Deming Yang, who have done the modeling to know with different parameters find in the histological study, find here. And as you can see, the results are very disparate. And in blue, you can see the carbon ratio measured in the chat canine, and the other are the modified parameter from the histological study. As so, the simulation show disparate results. The new model taking into account intra-tooth variation could be needed. Thank you for your attention.